Okay, here we are again with Mike 220 Virology 2. You can read the objectives here once again. We'll talk about bacteriophage T4 replication, replication features of a positive strand RNA virus, which is the largest virus family. Major features of a negative strand RNA virus here, we're going to use influenza. And then we're going to talk about retrovirus replication, and then a little bit about herpes virus, which is a double-stranded DNA virus, replicates in the nucleus. So we'll start small with bacteriophage T4. This should look familiar once again. We have a head with a nucleic acid in it, sheath and tail fibers. It's really quite a complex shape. It requires 20 genes, even though it is a bacteriophage and infects bacteria, it requires 20 genes. The capsid has linear double-stranded DNA, and the tail here has the fibers and the sheath and actually a base plate as well. So once again, as you remember from last time, the phage binds E. coli via its tail fibers, and attaches to the outer membrane a sugar receptor on the outside of the cell. And then the sheath contracts and injects the nucleic acid, the double-stranded DNA, single linear double-stranded DNA into the E. coli cell. And then what happens is early genes are transcribed, and you know that early genes are named like E1 and E2, things like that. And they're transcribed by host RNA polymerase. And the first proteins that are translated from that from the early genes are DNase and a DNA polymerase, because after all, with a DNA polymerase, we want to replicate the genome. So the bacteriophage makes its own DNA polymerase. And then the genome is synthesized by something called rolling circle replication. And I think you've already had rolling circle replication in your previous classes, either biology or Dr. Wang has mentioned it or talked about it in his lectures. If you haven't, go to the Norton animation websites, and there's a rolling circle replication tutorial video animation there that's quite good. And then late genes, remember, are structural. They produce the capsid and the tail proteins that assemble onto the membrane. Another late gene encodes something called lysozyme, and you already know that lysozyme degrades bacterial cell walls. So as the phage matures and gains its double-stranded DNA into the capsid and assembles, then lysozyme just degrades the cell wall, and the phage or the bacteria bursts, releasing the phage. So here is just kind of a review of rolling circle replication. So the first thing that happens is there's an endonuclease that nicks the circular DNA in this case, because even though the nucleic acid is linear, it can circularize because the ends overlap. And what happens is a primer comes in and starts synthesizing the one strand of DNA, displacing the other strand. So the secret here is that it displaces the other strand of DNA, and pretty soon the whole single-stranded DNA is displaced, and then the second strand can then be synthesized. The complementary strand can be synthesized on that displaced strand, resulting in. And then it can keep doing this rolling circle replication such that you have double-stranded DNA that's actually cleaved so that there's overlapping ends such that it can circularize, but this gets incorporated back into when the phage assembles, it can get incorporated back into through the sheath and into the capsid of the bacteriophage. And then lysozyme, then this is a mature phage, and then lysozyme can lyse the cell, the bacterial cell, and the phage can be released. So as you see, this is really kind of a even though it's a bacteriophage, a very complicated phage. There's 20 different products here that have to self-assemble and go together. And we actually know about this because we've been able to mutagenize some amino acids in the capsid and make giant head proteins, for example. So the phage size, the head doesn't know when to stop. It keeps adding these monomeric proteins into the capsid of the phage head. We make other mutants and we just get tail sheaths and no head. Now we have one that has a base plate and no sheath. So this is how we've learned to dissect viruses, and we started with bacteriophages, and we can do this now with mammalian viruses as well. So once again, this just talks about the replicative phage or the replicative cycle of the bacteriophage. Once again, gets in, makes messenger RNA from double-stranded DNA, and then the messenger RNA can then go ahead and make protein, as messenger RNA does, but from the double-stranded DNA, rolling circle replication makes more of the genome. So now we have proteins being made here from messenger RNA, early and late proteins, and we have genome being made. The genome inserts into the capsid, and as the phage assembles, it releases, this looks like it's coming out of a pore, but actually the bacteria burst. Okay, so now we're going to move into a more relevant system. We're going to move into sense RNA viruses, and the examples here are picornavirus. Picornavirus is or poliovirus is an example of a picornavirus. It's an enterovirus. It's transmitted fecal-orally. We've already talked about the history of poliovirus. It made as a matter of fact, one of the presidents of the United States, gosh, my history is not as good as it should be. I think it was FDR, perhaps, Franklin Delano Roosevelt, perhaps had polio. I think he was the one in the wheelchair. In any case, it is a positive sense RNA virus. Noroviruses, so if you think about noroviruses, you should think about cruise ships, because this is the virus that gives you diarrhea on a cruise trip, or cruise ship. So next time you take a trip on a cruise ship, you should worry about noroviruses. We're actually coming up with a vaccine, not we, but the field is coming up with a vaccine for noroviruses. And then flaviviruses, we've already mentioned, West Nile dengue, hepatitis C virus. We'll be talking a lot about hepatitis C virus here today. Rhinoviruses, these give you the common cold. Hepatitis A viruses are transmitted in food, so it's fecal-oral. And then coronaviruses, SARS, most of you may still remember SARS. About 10 years ago, we had a SARS outbreak. We'll talk a little bit about SARS in a subsequent lecture when we do epidemiology. So let's use an example of a positive strand RNA virus, okay, hepatitis C, bloodborne virus, okay. It infects about 200 million people worldwide, and there's a population that was born between the ages of 1945 and 1965 that is likely to be, well, a certain percentage of the population is likely to be infected with hepatitis C virus, and screening is recommended. The CDC recommends screening so that these people can take a new drug that's available that cures people of hepatitis C infection. And we can talk about that a little bit later. We had a hard time growing this virus for a long time. It only grew in humans and in chimps. So it was hard to make antivirals because you can't really do experiments in humans, and it's about $40,000 to $50,000 a year to keep a chimp, and some people don't think we should be doing research on chimpanzees. So it's very difficult to grow, and then finally some laboratories were able to develop culture systems, so cell culture systems. So they took hepatocarcinoma cell lines and figured out how to grow hepatitis C virus in those cell lines. And once you can you can grow a virus in a cell line. You can do lots of different things. You can add what you think are antivirals to the cell line and determine if the virus still replicates in the cell line. And that's easy to do because you can just do plaque assays, right? So we learned about plaque assays last time. And what you can do is you can add the drug to the cell that supports the virus, and then add the virus. And if your virus titer, or the concentration of virus that you get after 3 or 4 days is low, that means that the drug worked. If it's high, that means the drug, or the same as the control, the positive control, that means the drug probably did not work. Okay, so HCV is a very simple virus. It's like polio, very simple. It just has 3 structural proteins. Okay, it has protein C, which is monomeric, and we just have 1 protein forming the capsule. Unlike polio, where we had 3 proteins forming the capsid, with hepatitis C, we just have 1 protein, monomeric protein that forms the icosahedral capsid. And then proteins E1 and E2 are dimers, and they form the envelope of the virus. So that's what helps the virus, the receptor on the virus bind to host cells. Once again, it's positive sense RNA. It also has non-structural proteins. So 1, 2, 3 plus 7 is 10. So it really has very few proteins, and these non-structural proteins turn out to be critical for antiviral drugs because a couple of them are proteases, and we can target proteases with chemical compounds, small molecules. So once the virus gets inside the cell, viral RNA is translated by something called an IRES, or an internal ribosomal entry site. So it doesn't replicate really like a normal RNA. It doesn't have a 5' cap. It doesn't have a poly A tail. So it uses this IRES to attract a ribosome. And it makes 1 polyprotein. And that's a theme among viruses, is to make 1 polyprotein. And then proteases cleave that polyprotein up into the 7 non-structural proteins, E1 and E2, and then protein C. So if you inhibit a protease, you're going to be stuck with a big polyprotein that's kind of still stuck, glued together as 1 big protein, and the virus can't really form. So here is a cartoon of the structure. So monomeric protein C, and then E1 and E2, and then the positive sense RNA inside. Here's a picture of the IRES, or internal ribosomal entry site. And this shape here, so RNA can form shapes, DNA can form shapes too, but RNA forms this shape here, and the shape actually attracts a ribosome, and then a ribosome goes and translates this whole positive sense RNA, and it can do it because positive sense RNA is just like messenger RNA, and it makes this big polyprotein here, okay? And then what happens is these NS2 and NS4 and NS3 to some extent are proteases, and these proteases are kind of self-active. They are, even though they're in the polyprotein, they're active, and what they do is they go and they can cleave all the other proteins away from the polyprotein. So then all these proteins become functional, including an RNA-dependent RNA polymerase. So here we have a virus that's positive sense. And if it's positive sense, it can automatically hook up with a ribosome, translate a polyprotein. The proteases will cleave the individual proteins away from the polyprotein. One of those is an RNA-dependent RNA polymerase that can then go back to the positive sense RNA and make negative sense, okay, or antisense. And remember, antisense is a template for more positive sense. So you're positive, then you have to get negative, then you have to get positive again. I'll say that probably several times. Okay, one of the things the virus does in serum is it coats itself in low-density lipoprotein receptor, and as it binds to the cell, it not only binds it binds to hepatocytes, but it binds to a couple of receptors, CD81 and SRB1. Don't worry about that. I just want you to know the replication strategy of positive sense RNA viruses. You don't have to remember what the viral receptors are, the host cell viral receptors are. So we can kind of go to the next. So here we are binding, remember, but I do want you to know, remember attachment, and then penetration, and then uncoating. So here we have attachment, and then penetration occurs through tight junctions because cells come together. And so here we have penetration through what we call a tight junction. And now we have an endosome here, and endosomes fuse with lysosomes, right? And the pH of lysosomes is acidic. So what happens here is we get an acidic environment, and the acidic environment allows the endosome or the membrane of the virus to fuse with the endosomal membrane, releasing the capsid, which also denatures the capsid proteins and allows the genome to be released. Okay? This says exactly, pretty close to what I just mentioned. So now we have the genome released. Here is the positive sense single-stranded RNA, which is just like messenger RNA to a cell, except for this one, remember, has that weird IRES and recruits a ribosome. But that's okay. Recruiting a ribosome is exactly what the virus wants to do. So ribosome is recruited, we get a polyprotein made, and when we get the polyprotein made, the proteases are kind of automatically active, and then the proteases can start to cleave the individual proteins, including the RNA-dependent RNA polymerase. And the RNA-dependent RNA polymerase goes back then to this positive sense RNA, makes a negative sense or an antisense, and then that can serve as template for more genome of the virus, the positive sense genome of the virus. And that happens down here in what we call this membranous web. But what's really important, what I really want to call your attention to, is these proteins here, NS3, NS4, NS2 in some respects, and a couple of companies, as a matter of fact, have made antiviral drugs that inhibit these proteases. These are serine proteases, and we have serine protease inhibitors, boceprevir and telaprevir, and those two drugs inhibit the activity of the proteases that are kind of automatically active in the polyprotein. So if we inhibit those proteases, as I mentioned before, then those proteases can't go and cut out the RNA-dependent RNA polymerase. They can't cut out the structural protein or the viral receptor or the capsid protein. So really, once the virus infects the cell and this protease gets inhibited, there's nothing the virus can do. So the virus replication stops. And these are very effective drugs, as a matter of fact. They're used sometimes in combination with an older drug, ribavirin and interferon alpha. But these drugs are they're very effective and they're very expensive. They're about $80,000 a year, but it's a cure for people who have hepatitis C virus infection. So there's kind of a social argument right now on whether or not insurers are going to pay $80,000 to cure somebody, but after all, it is a cure. Okay, so this is a poliovirus replication, and I stuck this figure in. This is from an old version of your textbook. I put this in just because I like the way this shows. So once again, we have the attachment uncoating, so we have a positive sense here.